Hey folks, in this video we'll have a sneak peek into the history and discovery of microorganisms. The main focus will be on how great scientists such as Hooke, Pastuer and Leeuwenhock saw and discovered the invisible, that is the microbes. We will also look into the contributions of scientists which have helped our world enormously. So we'll have a small introduction and then the discovery of microorganisms and about the scientists Robert Hooke, Antonino Lewinhock and the spontaneous generation theory, Louis Pasteur and his contributions. So coming to the introduction, if we gazed at the night sky on a cloudless evening in a region far away from the light population, we will be amazed and perhaps a little humbled by the vast number of stars. It's hard to estimate just how many stars are out there. The Milky Way galaxy has around 100 billion stars. Now, if we turn our gaze back to Earth, we will find even more awesome abundance of microorganisms. So, what are microorganisms? Microorganisms are defined as those organisms which are too small to be seen by the unaided eye. The abundance of microorganisms is enormous. If we stacked all the viruses on Earth on top of other, they would stretch for about 100 million light years, which is about 43 times further away from the Andromeda galaxy. And there are at least 100,000 more bacteria in the ocean than the stars in the universe. And a teaspoon of soil has about another 100 million microorganisms. So this got me wondering, how did we discover microorganisms? Even before microorganisms were actually seen, some investigators suspected their existence and role in diseases. The Roman philosopher Lucretius and the physician Girolamo Fracastoro suggested that diseases were caused by invisible living creatures. The development of microscope was the first critical step in the evolution of microbiology. The earliest microscopic observations of organisms were made between 1625 and 1630 on peas and weevils by Francisco Stelluti using a microscope supplied by Galileo. Coming to another great scientist, that is Robert Hooke. He published the first drawings of microorganisms in scientific literature Micrographia in the year 1665. Here in figure 6 and 7 we can see his study of the flea anatomy. Coming to the second great scientist, Antonino Lewinhock. He was born in Delft, the Netherlands, and he is known as the father of microbiology and is one of the greatest microscopists and microbiologists. He was also one of the first to observe microorganisms using a microscope of his design, which was a prototype of the design discussed in Micrographia. Figure 9 shows drawings of bacteria from the human mouth which Leeuwenhoek observed. Leeuwenhoek earned his living as a draper and haberdasher, that is, a dealer in men's clothing and accessories. But he spent much of his spare time constructing simple microscopes composed of double convex glass. His microscopes consisted of lenses held between two silver plates that could magnify about 50 to 300 times. He illuminated his liquid specimens by placing them between two pieces of glass and shining light on them at a 45-degree angle to the specimen plane. This provided a form of dark field illumination whereby organisms appeared as bright objects against a dark background. So he observed these earthly particles that you can see in figure 10 and named them as animalcules. Figure 11 shows the Leeuwenhoek microscope, that is the microscope which he used. In 1673, he sent letters to the Royal Society of London describing his discoveries that he clearly saw bacteria and protists. In 1673, he observed the first ever bacteria to be seen by humans in water. He also laid the foundations of plant anatomy and became an expert on animal reproduction. In figure 12, you can see the anatomy of an insect wing. He also discovered blood cells and microscopic nematodes and studied the structure of wood crystals and also made over 500 microscopes to view specific objects. In figure 13, you can see the plant and feather anatomy which he has done in very detail. And his most important discovery was the discovery of the sperm. He described the spermatozoa of mollusks, fishes, amphibians, birds, mammals, and came to the novel conclusion that fertilization occurred when the spermatozoa penetrated the egg. In figure 14, you can see the flea anatomy, and the figure 15 is the page in one of his books. Next, coming to the miasma theory. So this is an obsolete medical theory which states that diseases such as cholera, chlamydia, or the Black Death were caused by miasma, that is, a poisonous vapor or mist believed to be made up of particles from decomposing material that could cause diseases and could be identified by its foul smell. This theory was proposed by William Farr, who stated that the inverse association of cholera mortality with the elevation above sea level confirmed the miasma theory as its cause. Here in figure 17, you can see a doctor wearing a particular kind of mask. So this was during the Great Plague in 1665. So here the doctors wore masks filled with smelling flowers to keep the poisonous miasmas out. And figure 18 shows a colored lithograph by Robert Seymour, which depicts cholera as a robed skeletal creature emanating a deadly black cloud. Coming to the theory of spontaneous generation. So this is the hypothetical process by which living organisms develop from non-living matter. The Greek philosopher Aristotle proposed that life arose from non-living material if the material contained pneuma, that is, vital heat. As evidence, he also showed several instances of the appearance of animals from environments previously devoid of any habitation. For example, the seemingly sudden appearance of fish in a new puddle of water. Then another scientist, Jean-Baptiste van Helmont, proposed that mice could arise from rags and wheat kernels left in an open container for 3 weeks. But in reality, such habitats provided ideal food sources and shelter for mouse populations to flourish. However, another person questioned Van Helmont's theory. He was one of his own contemporaries. This was an Italian physician, Francisco Redi. He performed an experiment in 1668 to refute the idea that maggots spontaneously generated on meat left out in the open air. He predicted that preventing flies from having direct contact with the meat would also prevent the appearance of maggots. So what Reddy did was he left meat in 6 containers, in which 2 were open to air, 2 were covered with gauze, and 2 were tightly sealed. And he observed that maggots developed in uncovered jars and did not appear in the gauze covered with the tightly sealed jars. Therefore, he concluded that maggots were formed only when flies were allowed to lay eggs in the meat. The maggots were the offspring of flies and not the product of spontaneous generation. Another scientist, John Needham, briefly boiled broth infused with plant or animal matter, hoping to kill all the pre-existing microbes. So what he did was, he sealed the flasks and left it for a few days. He observed that the broth had become cloudy and a single drop contained numerous microscopic creatures. Therefore, he argued that new microbes must have arisen spontaneously, but in reality, however, he had probably not boiled the broth enough to kill all the pre-existing microbes. Again, there was another scientist that did not agree with Needham's conclusion. However, he performed hundreds of carefully executed experiments using heated broth. So the debate over spontaneous generation theory continued well into the 19th century, and to put an end to this, the Paris Academy of Sciences offered a prize for the resolution of the problem. So at this time, a prominent French chemist who had been studying microbial fermentation and the causes of wine spoilage accepted this challenge. This was none other than Louis Pasteur. In 1858, Pasteur filtered air through a gun cotton filter, and upon microscopic examination of the cotton, he found it was full of microorganisms. He suggested that the exposure of broth to air was not introduction of a life force, but rather airborne microorganisms. Later, he made a series of flasks with long twisted necks in which he boiled broth to sterilize it. His design allowed air inside the flask to be exchanged with the air from the outside, but prevented the introduction of any airborne microorganisms as it would get caught in the twists and bends of the flask necks. If a life force besides the airborne microorganisms were responsible for microbial growth within the sterilized flask, it would have access to the broth, whereas the microorganisms would not. As predicted, the sterilized broth in his swan neck flask remained sterile for as long as the necks were intact. When the necks were broken, the microorganisms would be introduced, contaminating the flask and allowing microbial growth within the broth. Pasteur's set of experiments irrefutably disproved the theory of spontaneous generation and earned him the prestigious Alembert Prize from the Paris Academy of Sciences in 1862. In a subsequent lecture in 1864, Pasteur articulated omne vivum ex vivo, which means life comes only from life. This became the theory of biogenesis. Biogenesis is the development of life from pre-existing life. It is the opposite of spontaneous generation theory and asserts that living things can only be produced by another living thing and not by a non-living thing. The person who first came up with this term was Henry Charlton Bastian during the year 1837 to 1915. Some other contributions of Louis Pasteur include molecular asymmetry. He noted that the molecules occurred in two mirror image arrangements that could not be superimposed, and this laid to the foundation of stereochemistry. His work with silkworms is also very significant. He learned how to preserve healthy silkworm moth eggs and prevent contamination by disease-causing microorganisms. Pasteur effect. He discovered that the fermentation process could be arrested by passing air through the fermenting fluid, and this led to the introduction of the terms aerobic and anaerobic. His development of vaccines for chickenpox and cholera and also anthrax was his most important work. And the development of the rabies vaccine, which was a new inactivated type of vaccine which consisted of a neutralized agent rather than an attenuated microorganism. The germ theory of fermentation. So in 1857, he presented evidence that not all fermentation is caused by microorganisms and that specific microorganisms cause specific kind of fermentation. Another important discovery of Louis Pasteur is pasteurization. So he studied wine contamination and showed that it could be caused by microbes. So to prevent the contamination, he used a simple procedure. He heated the wine to 50 to 60 degrees Celsius, a process now universally known as pasteurization. Therefore, pasteurization is the partial sterilization of a product such as milk or wine to make it safe for consumption and improve its shelf life. So, coming to the conclusion, the history of microorganisms and the enormous contributions of various scientists has led to the development of the field of microbiology. It has a long and rich history initially centered in the causes of infectious diseases, but now it also includes practical applications of the science. There are various branches of microbiology, and one of the most upcoming branch is astrobiology, also known as exomicrobiology. So, modern microbiology reaches into many fields of human endeavor, including the development of pharmaceutical products, the use of quality control methods in food and dairy product production, the control of diseases causing microorganisms, and the industrial application of microorganisms. So, they are also used to produce vitamins, amino acids, enzymes, and growth supplements. These were some of the references that I used. So thank you all for your patience, and if you like this video, please do consider subscribing.